In this video, we are going to provide an overview on one of the lab experiments, which is the determination of caffeine concentration using UV visible spectroscopy, in short, known as UV-vis. We are from Group 22, and without further ado, let's get straight into the contents of the video. There are 5 main topics that we are going to cover today. So, let's get into the basic principles of UV-vis spectroscopy. Spectroscopy is the measurement and interpretation of electromagnetic radiation absorbed or emitted, whereas UV-vis spectroscopy deals with the recording of absorptions of radiations in the ultraviolet and visible regions of the electromagnetic spectrum. The famous Beer-Lambert law is actually made up of 2 laws, known as the Beer's law and the Lambert's law. In short, the Beer-Lambert law states that the absorbance of a sample at a specific wavelength is directly proportional to the molar concentration of the absorbing substance. First, let's talk about the radiation source. No single lamp provides radiation across the whole range that's required, so 2 are used. Normally, a hydrogen or deuterium lamp covers the ultraviolet range, and the tungsten filament covers the visible range. Next, we have the monochromator, which splits the radiation beam into its component wavelengths by the grating or prism, and then followed by a narrow slit. This slit ensures that the radiation is of a very narrow waveband. That is why it is called monochromatic. Next up, we have the sample cells, also known as the reference cells. It is convenient to have the sample in the form of a solution. However, one main thing about the solvent is that it should not absorb in the ultraviolet and visible range. The detection of radiation passing through the sample or reference cell can be achieved by a couple of photosensitive devices. Here is a general instrumentation setup of the UV visible spectroscopy experiment. Now, let's proceed to the brief background on the experiment. The main purpose of this experiment is to determine the concentration of caffeine in several commercially available beverages. They are mainly pressed coffee, instant coffee, black tea, green tea, as well as decaffeinated coffee. There are 3 things that are going to be investigated in this lab. First will be the maximum absorption wavelength of caffeine. Second will be the calibration using blank and standards of known concentration. Third will be the determination of concentration of caffeine of unknown samples with different dilution factors. Here are the 4 main objectives of this experiment. To determine the maximum absorption wavelength of caffeine, a full scan was done. As a result, the lambda max obtained for caffeine is 269.1 nanometers. A calibration experiment was done using several standards of known concentration. This is to obtain the calibration graph, which is the absorbance versus concentration graph. It can then be used to determine the concentration of unknown samples based on their absorbance units. To obtain the average concentration of caffeine in the unknown samples, simply plug in the absorbance values into the Y values to determine the X value, which is the average concentration. There are 3 sets of unknown samples with different dilution factors. Each sample consists of the 5 unknown beverages that we are about to determine. Here are the results for the sample without dilution. Moving on to the next topic, which is discussion, we will discuss the questions that are posed in the lab manual. Here are the default parameter settings for the method section that has been created by the lab technician. According to the Beer-Lambert's law, absorbance is directly proportional to the concentration of the analyzed sample. Therefore, in theory, if the Beer-Lambert law worked perfectly, a straight line would pass through the origin. However, the graph that we obtained does not pass through the origin. But since the R-squared value displayed is 0.9985, which is close to 1, this means that the calibration graph is approximately close to the theoretical fitted regression line which obeys the Beer-Lambert law. Therefore, the linearity of this calibration graph is accepted. I'm fairly certain about the accuracy of the calibration data. Why is that so? This is because the graph that we obtained displays the R-square value, which is 0.9985, which is relatively close to 1, and the standards prepared are very dilute, which means the Beer-Lambert law is obeyed. What if the standards were prepared at different concentrations? The calibration graph would be slightly different. This is because different concentrations would result in different degree of absorbance. However, this is only true if concentration of standards prepared are relatively low, as this is one of the conditions for the Beer-Lambert law to be obeyed. Let's take another scenario. What if the standards were prepared in high concentrations? The calibration graph would no longer be linear. The derivation of Beer-Lambert's law assumes that the molecules absorbing radiation do not interact with each other. However, at higher concentrations, the molecules are more than likely to interact with each other. Therefore, the assumption used to derive Beer-Lambert's law breaks down at higher concentrations. Based on statistical analysis on the experimental data, we have concluded that pressed coffee and instant coffee have the highest caffeine concentration, but we do not know for certain which one is higher. Same goes to black and green tea. We do not know for certain which one is higher. However, we are most certain about decaffeinated coffee having the lowest caffeine concentration. Therefore, in order to determine accurately the caffeine concentration for each sample, we carried out some research on various literatures concerning caffeine concentration, and the data we obtained is displayed in the table below. By comparing the data obtained from literature, we can deduce accurately the type of beverage based on the concentration obtained from the calibration graph. The beverage highest in caffeine concentration would be pressed coffee, followed by instant coffee, black tea, green tea, and finally decaffeinated coffee. The dilution factor only decreases the concentration of caffeine and its absorbance value. The amount of caffeine remains unchanged. This is because the type of beverage remains the same, so no, I wouldn't expect any discrepancies. One main source of error is the inaccurate preparation of the concentrations of the standards. The concentrations prepared might be too high, and this causes deviation from the Beer-Lambert's law. Therefore, the volume of the caffeine stock solution must be measured accurately. Here are some other common sources of error which will result in statistical deviation and some of the ways to overcome it. One main problem is the beverage samples may also contain other chemicals that absorb at a similar wavelength with caffeine, and this gives a high concentration of caffeine in a UV protocol. So how do we overcome this to achieve the most efficient analysis? First, we can repeat the experiment multiple times. In this way, we can determine if the data was just a fluke or represents the normal case. In short, it gives validity to the test results obtained. And next, we can confirm the validity by referring to the calculated standard deviation value. For example, we run 5 tests to obtain 5 sets of values of absorbance for the beverage samples, and then we use the mean of the 5 sets of data to determine the concentration of the beverage sample. We confirm the validity of the mean by referring to the calculated standard deviation value afterwards. This is the formula that is used for the standard deviation. The mean absorbance value is accepted as long as the standard deviation value is no greater than plus minus 2 standard deviation away from the mean. I would suggest HPLC and CE. Here is a basic instrumentation setup for HPLC. HPLC is simply a separation technique that is based on the distribution of analyte between a mobile phase and a stationary phase. Subsequently, the individual components of the sample migrate through the column at different rates because they are retained to a varying degree by interactions with the stationary phase. The substances are then detected by a suitable detector. Here is a general instrumentation setup for capillary electrophoresis. CE is a separation technique in which charged particles are separated based on their charge and size by their different migration rates in an electric field. A capillary tube is placed between 2 buffer reservoirs and an electric field is applied. A defined volume of analyte is introduced into the capillary by replacing one buffer reservoir with a sample vial. In short, HPLC and CE are the more suitable analytical method in this experiment compared to UV-vis. This is because they are suitable for identification, determination, and separation of the analyte sample, whereas UV-vis is only suitable for identification, and determination and not separation. The samples may be contaminated with other chemicals that absorb at the same wavelength to give falsely a high concentration of desired analyte in a UV protocol. HPLC and CE gives the possibility of separating those interfering compounds away, whereas UV-vis doesn't. Based on the literature data shown below, caffeine concentration obtained using HPLC and CE exhibit a very small difference. Therefore, quantification of both methods are very similar. Since we could not provide an accurate comparison of the quantification by HPLC and CE, let's move on to compare the analysis time, cost, and generated residues. Based on the literature data obtained, HPLC has a longer analysis time, more expensive reagents, and produce greater amount of residues. In short, the CE method, when compared to the HPLC method, presented a good relationship between cost and benefits, while enabling monitoring of the caffeine content in the beverage samples with the same reliability, with a faster and more cost-effective way. Yes, we have achieved our overall aim and objective. First, we managed to identify a suitable wavelength scan for caffeine, which is 269.1 nanometers. Second, we managed to obtain a linear calibration graph with the straight line equation shown below. Through the calibration graph obtained, we have managed to determine the concentrations of caffeine found in the beverage samples. We have also found out that samples with higher dilution factors have lower caffeine concentrations. Through this experiment, we have found out that pressed coffee has the highest caffeine concentration, followed by instant coffee, black tea, green tea, and finally, decaffeinated coffee. Based on our results, we can conclude that absorbance is directly proportional to concentration. We can carry out the experiment using CE or HPLC, as UV-vis is applicable for identification and determination only. It cannot separate the constituents of a mixture.